very much for coming today and to join this session. I'm joined by a very talented individual, Andrew Lesniak, who's going to share with you in the second talk. I'm going to give an initial talk kind of sort of my experience, and then followed by his talk. And then I think we're going to have plenty of time for discussion. This is kind of I think it'll be a very informal discussion. We can have plenty of time for questions at the end, unlike other Banff conferences. And I do thank you for coming, because I know there's a lot of competing concurrent sessions, so I really appreciate your attending. So I'd like to, basically I entitled this talk Banff Digital Pathology, and I'm going to discuss reproducibility and some morphometric methods that currently I've done and in the past I've done. And then talk about more and basically some plans that I think that the Banff is going to take next. And I do have a couple of disclosures, but they're not related to this talk. That have some funded, some research I've been involved with in the past. So I was actually, my first Banff was in 2009, and that was, I took a picture there then, and I hear it's going to be there the next time. The Banff Alberta Hotel, the Banff Hotel, the Fairmont. And then up the other one was actually, the conference where it was actually higher in the mountains. So it's a beautiful place. But Pittsburgh's been great too, and I know several of you have enjoyed it here. So the thing about Banff though is that there are a lot of scores, and I apologize it's so small, but that's kind of almost to illustrate the point that there are a lot of scores, there are a lot of parameters, and basically it's around 15 parameters, and if you figure about 10 seconds each for each parameter, that's about 150 seconds to score all that, which is about 2.5 minutes. But that's conservative, and we do a lot of other things. We count glomeruli and other things, and so that's why I put the sleeping person in the bed, because sometimes it's so much that it can put you to sleep. But the parameters actually go into, and again, these are small too, go into the diagnoses, and the scores actually are component points that go into the actual Banff diagnoses, and that's what we see here. The newest category was this chronic active cellular rejection for the kidney, but it has its own problems, and that's being discussed here, and challenges. And so I think it's going to be refined even more as you go through. But the difficulty is that there's variation in histologic rating, and this was illustrated nicely in this Farris paper in 2003. And in that paper, photographs and feedback were given to the participants. And that improved some scores, but not others. And even when the participants were shown photographs, the kappa values were still pretty not the greatest. And so when you think of a poor kappa being less than 0.2, fair being 0.21 to 0.4, most of those were actually on the order of fair to poor. And again, like I said, even after feedback was given, and even after the actual fields of interest were shown to the participants. This was an international scoring, mainly European participants. And so I think this illustrates the point that there needs to be methods to improve our assessment, and not replace the pathologist, but to complement the pathologist, and possibly help the pathologist screen the slides. And so that's how I got interested, essentially, is trying to assess biopsies in an objective way. And for example, I first started with the fibrosis score, and basically I wanted to be quantitative and ask how much fibrosis is there. Mainly because there are studies that have shown that fibrosis is linked to outcome. The most recent one that I have illustrated here was the paper that came out of the Banff on donor biopsies, kidney donor biopsies, and the regression, sort of almost like a hazard ratio for fibrosis, increasing fibrosis. If you compare like 6 to 25% versus 0 to 5%, was like 1.77. 26 to 50 versus 0 to 5 was 4. But actually neither of these were statistically significant. And unfortunately, the biopsies, were biopsies that were all transplanted because we were measuring outcome. So we didn't have any severe fibrosis in those biopsies. So none were greater than 50% fibrosis. So that could be why these logistic regression values were not statistically significant. But other studies have shown increased risk of adverse outcome. Dr. Randhawa, here in Pittsburgh, did a nice study in 2000, so like 19 years ago. And then Dr. Cerrone, who's also here at the conference I saw, Dan Cerrone in Barcelona, did a nice study. And he linked that to graft outcome of 1 year, and that was using morphometry. And obviously that was why I thought that morphometry could be good for fibrosis. But then there are other studies that have shown no predictive value. And I think this illustrates the fact that the sort of basically the assessment of fibrosis is quite problematic. And actually, as part of the, we conducted a trial, which I'm going to discuss a little bit more on some subsequent slides. But improved visual assessment, Dr. Chris Bellamy designed this, and he's actually here in the audience, designed this nice sort of pictorial diagram, sort of as a visual aid to sort of illustrate the increasing levels of fibrosis, both if it's sort of patchy, as you can see in this first sort of column here, and then if it's sort of more confluent, and that illustrates how much fibrosis is in these sort of little virtual biopsies. But and so that was quite useful and helpful as a visual aid. And he had done that, I think, before that for like steatosis in the liver. But this was actually helpful for our participants in a study, which I'll discuss later, in assessing fibrosis with the visual assessment. And that was quite helpful. And because there are essentially two ways of assessing fibrosis. Sort of like the computer assesses fibrosis, if you sort of look at a biopsy and say, how much is fibrous tissue? And sort of think almost like, as I'm going to discuss later, a positive pixel count algorithm. How much of this area is fibrous tissue? Another way that I think a lot of pathologists actually assess fibrosis is to say how much of the biopsy is abnormal, how much of the parenchyma is actually abnormal. So that's the second column here. And in in a study that I did with Bob Colvin in 2000, we published in 2011, we did that in one center, and then we actually extended that to the Banff, which I'll show in a subsequent slide. Just when we did it in one center, with Bob Colvin and Neil Smith, who's also here at the meeting, two people I trained with at the time. Basically, we were all proportional. I was the third observer; they were the other two, and the regression values were quite good. Typically, when you look closer at the numbers, and I know there's a lot of data there, but your cutoffs, pathologist cutoffs, tend to be a little bit less. So your absolute sort of dynamic range of fibrosis that you would assess on a biopsy might vary, but there is proportionality here, and the R-square values were like 0.7 to 0.99 amongst observers, and also using the two different ways of assessing fibrosis. But we knew that if we extended to multiple institutions, it probably would break down, and that truly did happen, and I'll show you when we did the larger Banff trial. But so we were thinking about ways to improve this better and make this better. And at the time, Dr. Colvin had a whole slide scanner that he just had obtained in the laboratory. So we decided to use the whole slide scanning to assess both intra-observer and inter-observer variability. And these scanners, obviously, as you know, and many of you have these now, scan the whole slide automatically, and they can scan multiple slides in a batch. The slides can be quite large. If it's a large piece of tissue, they can be 1 to 2 gigabytes. Biopsies are typically smaller, a few hundred megabytes typically. And so basically, they scan the slides automatically, and you can assess things quite usefully and share it with multiple individuals, and you don't have to worry about breaking slides if you share multiple sites. And so there are a lot of advantages with whole slide scanning. And we decided to do a fibrosis quantification trial. So we stained the biopsies like serial sections with a lot of different stains as illustrated here on 2 days, and we compared a day to day, and actually the agreement was pretty good. I don't think I'm going to show that data in this slide, in this presentation, but the days correlated quite nicely. And also, there were correlations amongst the stains, and I'll show that in the subsequent slides. And so we did collagen 3 immunohistochemistry, PAS, and trichrome, and Sirius red. So Sirius red's a nice stain, and a lot of people have used it in the past for research. But the difficulty about Sirius red is that it's seriously red. It's like Mark Rothko's Red on Red. You know, Mark Rothko, the painter who did these paintings, and one of them was called Red on Red. So Sirius red's red, and to get a good assessment of fibrosis, a lot of people claim that you need to do polarization. And actually the whole slide scanners don't do polarization. So for our study on fibrosis, we had the help of Dr. Paul Grimm at Stanford, who's done a lot of work. I think this went all the way back to when he was in Canada with Dr. Nickerson and Rush. They've done a lot of studies on using Sirius red for quantitating fibrosis, and they've done some nice papers. He's mainly a pediatric nephrologist, and he has a big interest in digital imaging too. And he's done morphometry and has related that to immunosuppression in a variety of papers. And so he was nice enough to help us with our quantitation to see how the measurements correlated. And then we thought of this method, what we call the T minus P method, trichrome minus PAS, because trichrome will stain basement membranes too. It will stain glomerular basement membranes, it will stain tubular basement membranes. So if you use the PAS, which stains the basement membranes a hot pink, you can subtract out the basement membranes, we thought, using this approach. And I call this real intelligence, because it's not artificial intelligence like a lot of people, and I'm going to try to discuss later, but sort of using real intelligence to try to think of better methods of quantitating fibrosis. And so when the quantitation is conducted with the positive pixel count algorithm, you get what's called a markup image, which actually highlights areas of interest that you have quantitated. And actually, usually the algorithms are quantitated, or actually come out of the box ready to quantitate immunohistochemistry. So you can actually change the hue to detect fibrosis or the pink of the PAS or whatnot. But it works quite well for collagen 3 immunohistochemistry also. And with this study, we also correlated things to function. So, the stains actually correlated quite nicely, and I'm not going to show that data in interest of time, but we also correlated it to function. And the things that correlated the best were actually collagen 3 immunohistochemistry and Sirius red, actually the unpolarized morphometry that Dr. Grimm had done. And the collagen 3 versus GFR, the R squared was 0.5, so it's not perfect, but it was statistically significant. And for visual assessment, though, was essentially almost as good as the morphometry. So it still shows that the pathologist, the eyeball test, essentially came out equivalent to what the computer was doing. So it still showed, at least with these studies, that the pathologist still had a job, essentially. So they were still doing as well as the morphometry. So after this, we, in 2009, at that meeting that was the last one in Banff, we did a pre-meeting survey where we assessed practices with regard to stains and section thicknesses, highest and lowest of fibrosis that was reported by the pathologist, and the assessment approaches. And we had initially in the pre-meeting survey, we actually had 250 participants in a Survey Monkey. It was mainly organized by Dr. Colvin. And we reported that in the later paper that where we actually asked the pathologist, and this is shown here, there were about 27 pathologists who assessed some slides that we scanned and made available. And visual assessment, though, was across the board. Each one of these columns essentially is a case. So you can see that the fibrosis reported by in the sort of bar graphs were the percent fibrosis reported by the pathologist. And you can see there's a wide range of fibrosis reported by the pathologists, unfortunately, in this study, and it does highlight that there's a great deal of variability. You know, the biopsies are ordered by the in order of the increasing collagen 3 immunohistochemistry, morphometry, and then the GFR is shown with these little triangles. And collagen 3 immunohistochemistry did have a weak correlation with GFR, as you can see here. And it actually, so the GFR is shown in this lower column here, lower row here. And the GFR versus collagen 3 immunohistochemistry was 0.44, the R value on that. Now, the approach by the pathologist actually was using approach B, which is the percent abnormal, actually did almost as well as collagen 3 immunohistochemistry. So that, again, shows that pathologists are almost as good, at least in this study, and likely more things need to be added before fibrosis, added with fibrosis to eventually give a better correlation with GFR if we use computerized morphometry. And so there was a wide range of variation. Another thing of note with this study, though, is that the kappa values were quite, unfortunately, quite poor. So as you can see, for all the approaches, they were almost between 0 and 0.1. Another thing assessed, though, in this study was the TI score, the Banff TI score, which is the total inflammation. And the kappa for that was 0.5. So it does show that, obviously some things in the study the pathologists did pretty well on, but unfortunately fibrosis, for whatever reason, pathologists were not doing quite as well. We did look at, have a couple of different individuals. We had some people in Alberta. Michael Mingle helped me a great deal with this study. And some people, Benjamin Adam, who is now in Alberta, and I think it was at the time also. He measured the fibrosis the same way I did with the collagen 3, and we correlated quite nicely. So it did show that the morphometry was reproducible, but unfortunately the pathologists were not. So what about, so that's fibrosis, which is, osis just refers to a state of a tissue or of some condition. But what about itis, the inflammation? And there are a lot of contributors to inflammation. This is from a figure that Dr. Coleman and I did on fibrosis, as sort of a review article. There are a lot of things that contribute to fibrosis: mast cells and macrophages, lymphocytes, and dendritic cells. But lymphocytes can be assessed quite nicely, and so I showed this and to talk about the itis of the inflammation, and so we can basically measure inflammation easily with immunohistochemistry, and that's shown in this sort of study that we did. Someone, Andrew Smoon, who was a resident at the time, he's now moved on, but he was a resident with me. He actually sort of came up with some ImageJ algorithms in addition to the positive nuclear algorithm that Aperio offers to actually count the cells in a biopsy, and essentially use flow cytometry on a slide. I know we're going to see much more sophisticated studies conducted by the Pittsburgh group. This is a simple study just to show that there are a variety of ways of counting cells, and this is just a simple one using CD3 immunohistochemistry that we were able to do. And we related it to the severity of rejection and other things. There are other algorithms that can be used. This is what's called a microvessel density algorithm, which can actually sort of count vessels and the density of the vessels, and also measure the size of the vessels, the wall thickness, and other things. And so we did a study, I don't know if I saw her at the meeting, but Gazim Toomer, and who was a HLA fellow at Emory at the time, and also Tiffany Roberts-Wilson, who's also at the meeting. They're now HLA lab directors. But with this, we were actually relating the microvessel density to the mean fluorescence intensity of the donor-specific antibody. And we showed that there was a weak correlation with C4D immunohistochemistry and microvessel density. The C4D microvessel density, and the MFI of the DSA, but actually the human sort of C4D score, the 0, 1, 2, 3, C4D 1, 2, 3, actually did better than the computer. So again, this is showing that humans, and there's more refinement that we need to do with the computer. So we actually never published this, but we did present it at some meetings. But in a sense, though, we should have, because it actually showed the pathologist, again, still had a job. This morphometry can be done in other organs. And this is from a paper that a resident at the time, who's now attending at Columbia University, Michael Lee, did. He's a pathologist now at Columbia. But at the time at Emory, he was a resident, and he helped me with a steatosis morphometry project. And in this, we quantitated steatosis in biopsies. And actually, we had collaborators, Dr. Diego Martin, who's now in the University of Arizona. He's department chair. And we also had a very great pediatric hepatologist, Marion Voss, who does great studies on steatosis, also with radiology, measuring steatosis with radiology. So with this study, we were able to show that the computer correlated with radiology measurements of steatosis, and also the pathology assessments of steatosis. And there were other things like body mass index that we had weak correlations with. So it was kind of, we were able to show correlations with the computer in humans and a lot of other parameters, which was quite useful. So, there's a lot of talk about machine learning and artificial intelligence now, and we have tried to do that in our group, and we're now doing more, which I'm going to discuss a little bit in a couple of slides. But this is one early thing that I tried to do. I showed the Genie movie there, because this is what's called the Genie algorithm that Apereo offers. And you can actually train the Genie algorithm to recognize different areas of biopsy, such as glomeruli, tubules, and vessels. And so, and you actually circle the areas, and then train the algorithm to recognize different areas. So you can see that basically you can train it to recognize glomeruli and tubules. It's not perfect, but it works. And you can, but when you, so it allows segmentation of the different areas, and then you can measure, for example, you could layer it with the CD3 immunohistochemistry and detect how much infiltrate is in glomeruli versus interstitium. Or you can even add, with this study I added on vessels. Now the accuracy of the detection does go down as you add more areas, and that's why more training is needed. And so at the time I was trying to train it more and more, and you can increase the iterations. It actually has sort of like it's been explained to me, it has like an internal database of textures. And so it combines those. It's almost like satellite imagery that our government uses to recognize bomb silos and things like that. And so you can increase the number of iterations that it compares. But the thing is as you increase the iterations and also slides, the sensitivity and specificity does go up, which is great. You can see it starts to go down as you add more slides and keep the iterations the same. But then as you add more iterations, the sensitivity, specificity, and sensitivity for both glomeruli and tubules goes up. But unfortunately, as you increase the iterations, the time for analysis, and unfortunately we had it almost on a virtual server with our bioinformatics group, and it was, and unfortunately eventually the server died. So, but you can see that the time does go up, but more refinement is needed. And so the great thing about this, so this was one of my early sort of attempts at artificial intelligence and machine learning. But now we actually have a nice project going on in Emory, and actually Julian Hogan is here in the audience. He has a grant, which is I'm really excited about, that we're going to be doing at Emory. We have a group of individuals, PhD computer scientists, Lee Cooper and David Gutman, we're putting it together. Julian is a pediatric nephrologist who's visiting from France at Emory for several years, and he was the mastermind of this project. I give him all the credit. But basically what we're going to be doing is collecting a cohort of pediatric transplants from multiple centers, and then applying artificial intelligence and machine learning algorithms to these biopsies and to whole slide scans of the biopsies. And we actually already have a project underway that we're doing that, and hopefully going to be getting some results soon. So that's some of the approaches. There are new things coming out and sort of novel microscopy techniques that I'm going to mention here just briefly. One of the first ones I'd like to mention is this digital spatial profiling, which allows you to there's a lot of talk here at the meeting of the nanostring, obviously this new BHOT sort of panel of nanostring probes has been released that has like about 700 genes for a variety of things, rejection and infection and other problems, mainly in kidney allografts, but I think it's being used for other allografts also. But you can also localize those derangements in biopsies, and nanostring offers this thing called digital spatial profiling, which allows you to capture with tagged antibodies, capture areas of the biopsy, and then do the nanostring analysis on different areas. And you can see how, get a spatial relationship of how the biopsy is actually, the molecular derangements are occurring in the biopsy. So that's one thing that I think is going to be quite useful in the future. And I know that Dr. Michael Mingles is basically one of the biggest leaders, and also, as I mentioned, Benjamin Adams done a ton of this, and also Bob Colvin and Neil Smith have done a lot of nanostring. So I think they also have efforts underway to do the digital spatial profiling. There are other techniques, spectral imaging, which allows, you can even do imaging of slides that haven't even been stained. And you can discriminate closely overlapping spectra and emission spectra. And for example, quantum dots or chromophores or fluorophores. I know maybe Andrew's going to talk about this, but I know Dr. Dimitris has worked on quantum dots in the past and has now moved to other things. We also have tried to work on quantum dots in the past and found that they had some issues, and so I don't know if we have time to get into that, but we've had issues with the quantum dots in the past too. But we had a similar instrument that we've worked on. There's this new instrument, the Vector Polaris, and I see Dr. Stevenson is in the audience, and she's really pioneering this. You can stain for multiple markers at once, and and do a lot of different things. And then there's such thing as second harmonic microscopy. I know that Agnes Fogo, who's here at the meeting, has done this with her group at Vanderbilt. And you can, basically you can image even animals apparently. I don't have a lot of experience with this, frankly, unfortunately, but there are things coming out that you can allow. And you can also visualize unstained, as I mentioned. There are ways of visualizing unstained slides. And this is useful for, if you image the slide before you stain the biopsy, you can measure certain things before and then stain. You can have an image of the biopsy before you stain in that way. You can get certain areas. This is another approach, FTIR. I think this is a group from France who does great work in that. I've heard them present this work, Fourier transform infrared. And again, this can be done on unstained slides, and so I think this is useful if you're going to, if you want to do some imaging before you stain the biopsy. This is one approach. This is another infrared imaging system that's been done. So just the references are there, but I don't have time to go into that in detail. But it's there for you if you want to take note of that. But why do we do all this? And I'll end this here. Is why do we do all this morphometry or try to apply machine learning or artificial intelligence? Well, I think of pathology, and we were talking about this earlier, is essentially the ground truth. Or, I've heard people say, pathology is the gold standard, and I like to think of it that way. Other people have said, oh, it's the bronze standard. It isn't quite. But I think, there are ways of refining pathology, and even though I present, problems with the inter-observer and intra-observer variability. There are ways of improving that. Pathologists can be trained, and also computers can help. And I think of another thing that AI stands for is augmented intelligence. We can use the computer to augment our intelligence, and not just artificial intelligence. Because all of these omics measurements, such as fibrosis, there are a lot of omics measurements. Osis is a state like fibrosis, but this can be related to omics measures, such as genomics, proteomics, inflammasomics, or kinomics, antibodies, and then also eventually pathomics, which is actually sort of, Bob Cole was the first person I heard who called something pathology, pathomics, and I really like that term. And eventually, these can all help us understand mechanisms. And then they can also be related to clinical parameters. These measures are more reproducible, potentially. The eye is nonlinear and semi-logarithmic, so that's why humans have issues. And so I think that's why automation can help, but augment what humans are doing. And as I mentioned, this quantitative data can be related to treatment dosage and outcome and response, pathogenesis, and eventually, like as I mentioned, help understand mechanisms. And also can help, this is from a review article I and some others at Emory wrote. And basically, scan slides can be quantitated. You can select areas and also do artificial intelligence. But this can also be integrated into reports. And as others have mentioned, you can have a toolbox of measures, as I illustrate here. But eventually, some of these things can be quantitated even before the pathologist sees the slide. So some of the report can actually be auto-populated before pathologists even look at the slide, and can help provide, sort of part of the report and reproducible portions of the report, and then the pathologist can focus on other areas and free themselves up to do other things. So right now I can see that clinicians assemble gestalt qualitative assessments, and they assign therapy. But in the future, I think that pathology analysis and toolboxes, either real or artificial, can help pathologists, in AI interpretation, or percent fibrosis, percent cellular infiltrate, C4D staining, or cellular immunophenotype and co-localization, and can help the clinician really have a quantitative report that can be used to assign therapy. And since, one of my prior colleagues, Randy Henniger, used to call pathologists, especially renal pathologists, armchair nephrologists. We're usually sitting in our armchairs looking at the slides, and we actually help the clinicians. But so we'll even be more of a part of the team, I think, if we can provide even better data for the clinicians. So I think, what are some next steps that Banff can take? And I think that we're going to have hopefully a white paper coming out after the meeting that Dr. Kim Solis has helped pioneer. He's sitting in the front row here, and I've been working with him. Julian Hogan's been helping, and others to eventually assemble a community of individuals willing to support and collaborate on digital pathology, and there's a variety of things we could do with this. So I think initially we're going to survey the current state and figure out who's using scanning of slides, who's using artificial intelligence, eventually survey the current state. And another project that eventually I think is going to take a while to develop, but develop an image bank for testing of algorithms. And the reason why I mention this is because there was a nice presentation at our United States and Canadian Academy of Pathology this year, where the chameleon trial for breast cancer was presented. In that study, these lymph nodes with metastatic breast cancer were uploaded to a server, and then groups could actually test their AI algorithms on this bank of images to see which group did best for detecting like metastatic breast cancer, for example. And that was a yes or no, But I think that, if Banff could do this, assemble an image, a bank of images, I don't think it's going to be as much a yes or no. Some algorithms are going to be better than others at detecting things like fibrosis or inflammation, or, I can eventually, with our algorithm, we want to eventually have a virtual Banff score, but I can see how different groups are going to be better at other things than others. And eventually you want to predict prognosis, and so hopefully we can have really nice, phenotype data along the lines of Alex Lupe, that he's assembling a nice sort of data structure, and eventually we can have all that data. And maybe we can even have official Banff algorithms in the future, such as targeted algorithms, such as what's known as sort of, I've heard Dr. Demetrius call them handcrafted algorithms. I like to call them real intelligence algorithms, that you're actually, the pathologist is thinking of things to quantitate, such as fibrosis or inflammation or steatosis. Or you can have artificial intelligence, the Banff artificial intelligence or machine learning algorithm. And you can also have, hopefully, data pipelines for integration of the omic, with omic data, such as along the lines that Alex Lupe and the Paris Transplant Group are doing, and even integrate maybe other things. He had a great presentation on natural language processing. So that's what the Banff, I think, is going to be doing with the with the working group on digital pathology, and also the white paper. So I'd like to thank a lot of individuals at Emory, and also my prior institution when I was at Mass General in Boston. That was over 10 years ago now, but I still, really think about my time there a lot. And also Michael Mingle, who helped a lot with the Banff, and also the people who, many of you may be in the room, who participated in those Banff, in the fibrosis survey, the Banff survey, and we're going to be sending out another one on digital pathology. So I really appreciate those participants. And also, thank you for the invitation for this speech. Thank you for inviting me for presenting this. And also I have collaborators at Emory and Duke, great surgeons so I thank them a lot. So, and there's some references of some things I've done, and I thank you. I don't know if we want to have question time now for me, or I guess we have time. Or,
yeah, we have a pretty relaxed.
Yeah, it's relaxed. We're gonna have, Doctor. We're gonna have plenty of time. So I know Dr. Demetrius is here, and a lot of other people who have tons of expertise in this area, Andrew Lesniak is going to have a great presentation, but we have time for questions now, and then we're going to have time, his presentation, then time, I think, at the end.
Just go through the two quick presentations and have it open.
Okay, yeah, we can do that. Yeah, so
we can do that. Sure.
Yeah, if you want to go ahead and yours, yeah, and then we can do that. Yeah, we don't have any attrition of the audience, so so thank you for that.
Thank you, Dr. Farris. Very, very informative. Looking for the F5. One of the things I noted about getting older, even though I guess I'm still fairly young, is that I can't see quite as well with glasses on when I try to read up close. So I tried to do my professorial glasses today to make myself look a little more intelligent or bright-eyed, but hard to see the keyboard. Well, thank you to the Banff Working Group, also to Dr. Demetrius and my fellow lab colleagues. Without their help, none of this could be done. Jake especially, really believes in this aspect of digital morphometrics. I struggled a little bit for a slide or for a title to this because there's so much material here in this transplant digital imaging space. There's so much material that is applicable to this field of pathology as a whole, from the advancements in digital imaging. And Dr. Farris did a very, very nice job at summarizing a lot of that. And so I was going to sort of focus this on the technology and algorithms at work. Dr. Demetrius has reviewed a lot of the data that we have generated out of our group in Pittsburgh at various sessions here and through other presentations across the industry at various meetings and the like. It's a very eye-opening for a computer scientist such as myself. It's a very eye-opening experience as to how much information can sort of be unlocked from a visual image. whether you use advanced artificial intelligence features or whether you use handcrafted features. As an aside, we didn't coin that term. I think, I don't exactly know where that term came from, but I've seen that referenced in other places. So we use it. I think Jake uses it, but I don't think it's ours originally. But it's just an amazing amount of a wealth of material. Now, one of the things, a little aside, that I wanted to bring up here was to talk about optics. And it goes back to my roots as being tied to the development of digital whole slide scanners many, many years ago. But one of the things we found very quickly in Pittsburgh, especially as we look to both transmitted light, traditional bright field imagery, but also as we've adapted our techniques to include fluorescence, is that there's always discussion of magnification and there's always discussion of resolution. We know what the terms mean empirically. Magnification is how big something is, and resolution is how delineated you can recognize a structure or how crisp something is to our individual eyes. So in the whole slide scanning industry, you get a lot of crosstalk between people talking about, is 20x resolution good enough? Do you need 40x? Well, the 40x can be looked at in a number of different ways. You can have low quality 40x lenses and you can have higher quality 40x lenses, aka resolution. So the numerical aperture of the lens and the overall optical system as a whole plays a huge part in terms of how far digital analytics can take us. And I think you probably agree. So the numerical aperture is in essence how much light a lens can collect. The more light you can collect, the more resolution you can get. So if you have a low numerical aperture lens, you're seeing much less than if you can look at something with a high numerical aperture lens. And I know that Jake has showed this slide, and it may be a little less, the point may be a little muted here on the screen and blown up. But on the top is an image of stop on the PowerPoint. How about that? We have to do this. We have to start that again. I'm sorry about that. Looks like PowerPoint crashed. So the top image is something captured at 20x at a 0.5 numerical aperture, and the bottom image is something captured with 40x, but the critical component is the 0.95 numerical aperture lens. The relative resolution based on this particular scanner worked out to be about 0.34 microns per pixel for the top and at about 0.16 microns per pixel at the bottom. So a much, much higher resolution, but when you start getting into advanced morphometric analysis, the more data and the more detail that you can present to algorithms, the higher the sensitivity, the higher the accuracy of this data overall. So I think it's an important point to consider that, as most of you probably do have digital scanners in your tool sets. Some are older, some are newer, some are the new types of imaging techniques that are coming out. But one thing to consider when you do talk to vendors and you do start to explore this, and perhaps even for the white paper considerations, especially as you get into renal pathology, Dr. Solis, the challenge there is to make sure that you are equipped with the right resolution to make an effective diagnosis, but also to enable those downstream digital algorithms. One of the challenges of this, and why many of the vendors, when the field was early, had gone to a lower resolution, is it makes focusing and many other things problematic. So the challenge becomes, as the resolution increases, the depth of field of the objective lens decreases. So you have much more of a challenge in terms of how to focus and how to capture this information reliably for a digital scan. The nice thing about computing technology, a lot of people will quote Moore's laws that things tend to get twice as fast and half as much over 18 months, is that computational power has increased, the amount of information that we can get from a digital camera quickly to a PC or to a Mac has also increased, so that algorithms that are able to process multiple focal planes during a scan and collapse all that information into a single field have become much more prevalent. And in fact, and again, not to look at specific vendors, but I know for a fact that 3D Histech, their new line of scanners, they call them the flash systems, exclusively use multiplane capture when they're actually imaging, because they are imaging at such a high resolution and they have to do that to maintain an accurate focus throughout the specimen. So just for the audience as a whole, for those of you who have not perhaps invested in digital pathology or scanning technology, it's something to consider and to keep in the back of your mind. Other tools, it's very interesting to watch this field evolve, because there's been a number of tools now that have come out to make the overall workflow possible. So I know within our lab, we do have quite a rigorous QA process. We end up with a small percentage of rescans, not as high as most people, probably in the range of 5 to 8% for bright field scans. But there has been quite an effort to develop and use artificial intelligence methods to increase the overall productivity of the workflow. So using these call them efficiency tools or call them histo-QC tools. There's a couple of well-known ones. I think it's Matabushi had histo-QC, which is an open source platform available to the world for looking for artifacts, folds, out of focus representation areas and things. And then there's been another from a group at Google, I guess it's Google AI Healthcare. Kohlberger has also produced an AI-based tool to help ultimately increase the throughput and minimize, how much rescanning and whatnot has to be done. So ultimately, if you can make this more practical, and if you can eliminate, errors or out of focus data getting downstream to the pathologist or to your machine vision research team, the better you be, the better you're going to improve your data. So one of the things that we rely on heavily within our work, and again, you've seen Jake's talks, is fluorescence. I think WSI, whole slide imaging, with respect to fluorescence, has really made a tremendous impact on the field. As most of you know, fluorescence is just the basis of applying the fluorophore or some agent that will collect photons and then emit photons at another energy level that can then be captured by a camera. Digital cameras have they have always been many, many more times sensitive than the human eye, just because of the opening, the time of exposure, the dwell time that you can give to a signal. However, in today's day and age, given the 16-bit CMOS cameras, cooling and whatnot, the sensitivity is taking a logarithmic step forward. So the ability for a scanner to capture very, very weak signals with a high degree of signal to noise is actually very practical in today's workflow terms. So fluorescence is something that I don't think that a lot of people have adopted to. I think the devices have been few and far between that have done it well, but there's tremendous benefits to using fluorescence whole slide imagery. Once something is scanned, it's preserved digitally and it can be revisited and or distributed as needed. Ourselves, we've started looking into scanning of native kidney panels, potentially for clinical sign-out purposes. Having the ability to capture fluorescent C4D and the various other stains that are part of the panel is absolutely critical to that. But one of the things I didn't put up here, and we like to talk about it amongst ourselves, is that it becomes a living data set. So once the image is captured, you can always go back and revisit that fluorescent image to try and extract more and more information. As you learn something about the pathology or the morphometrics teaches you about patterns that may exist, you can go and you can pull that data up again and reanalyze it. Second thing is the scan is performed at a higher magnification, or AKA a higher resolution. Therefore, you're capturing many, many more photons. So what you can see with your eyes when looking through, let's say, a 4x lens on a fluorescence scope versus what you can see with a digital equivalent of a 4x lens, quote unquote, from a fluorescent scan is tremendously more powerful. And we were actually talking, I don't think Dr. Farris was there, but earlier, is that I think it ultimately changes the phenotype of how a pathologist could use a fluorescent image, because so much more information is available to you at a low power. You almost retrain yourself as to how you actually look at that slide, and you gain trust in that image as you're looking at things at a much, much scaled down view. So it overall increases the efficiency. And then last is, to us, it really enabled the use of image analysis, because now you can apply N fluorophores, you can stain for multiple analytes together, and you can combine all that information into this quote-unquote soup and then tease out various types of structural and spatial relationships with that. Without the ability to do a multi-channel fluorescent image, it would be a challenge to do, with some of the work that we've done with Dr. Feng, Alberto, and colleagues, just because we don't have that ability to multiplex. So that's been a huge, huge step forward. Last and not least, I think, fluorescence used to be a very expensive proposition for a lot of people. Bulbs are costly. It was a very tedious system to set up. You had to have a lot of domain knowledge explicitly about fluorophores, hardware setup, the way you quote-unquote build the recipes. get an accurate and usable scan. But I think there's been a lot of technology improvements, and probably LEDs are one of the biggest, because we've sort of moved away now from metal halide bulb sources, and we've gone into an arena where you can excite at very specific fluorophores. You can combine the LED technologies to create a very wide bandwidth. And if you look at the graph behind me, the upper panel, the traditional bulbs present data at multiple peaks, or excitation light at multiple peaks. What happens is with LEDs, you're only exciting one individual single area. So you're increasing your overall sensitivity of what you're able to detect. And then the cost effectiveness has gone through the roof, quote unquote, simply because bulbs, which may get you 500 to 1,000 hours and may cost you $1,200 per bulb, if you multiply that over the number of fields that you're capturing when you're scanning a whole slide digital image, you may end up burning through 4, 5, 6, 7 bulbs or more a year, depending on your volume, if you were actually going to implement this in a workflow. With LEDs, the lifetime is thousands and thousands of hours. So the cost-benefit ratio of incorporating fluorescence and the overall cost to maintain it and keep it going has dropped tremendously. And then last, there are a lot of new filters and new optical coating techniques that enable what we call multi-band capture. So instead of capturing one fluorophore and waiting and capturing another, you can actually use the LEDs to dynamically change the wavelength at which they excite, and only have one filter in place to capture multiple fluorophores at the same time. So it's dramatically increased the scanning time. One of the things that again, Jake's presented on a lot with the data is we call it the tissue tethered cytometry, or you'll see the word near sight. And that's a software system that we've developed for our use in Pittsburgh, and it's also available for collaborators who want to use it. The reason we did it was not because it was a not invented here type of question, or we thought we could do something better than the others in the industry or the vendors. It was just so that we had control over the ability to change the software or produce data in a way that was more amenable to the research questions that we were asking. But one of the biggest challenges I think that when we sat around the table and discussed this many, many years ago was we wanted to make a system that was as reliable as possible and could be run on multiple slides and produce a result that was reproducible. So when you think of fluorescence, many people talk about thresholding and in essence manually choosing variables. So you apply a lot of sophisticated artificial intelligence to a downstream product that potentially you're altering upstream by changing values or setting things based on a human input. So we wanted to make the system as automated as possible so that it was based on variables that were not dependent on human threshold settings. So we sort of came up with this concept that we like to talk about and call probabilistic segmentation. And in essence, it's based on what are called Laplacian of Gaussian kernels, or hat-type filters, which allow you to set a measurement that says, I want to express something and I want to know that a pixel is on or off based on a difference from background. So it's a simple concept, but it enables you to be less threshold dynamic or less threshold specific when actually setting up to analyze these images. The second thing it gives us the ability to do is to define the field shape. And what I mean by the field shape is you may be looking for objects that are more circular at times, you may be looking for objects that are more elliptical, you may be looking for abstract shapes such as portal tracks. So it gives you the ability to combine all this type of information using a Bayesian type of system, whereas you know the likelihood that a particular cell or a particular pixel will be part of a group. You can combine the intensity data that you have with information that you've gathered about the slide based on these filters, run them through Bayesian logic, run them through the probabilistic segmentation at that point, and come up with a probability that a pixel is part of a group or not part of a group. In this case, groups represent objects. So what we're trying to do is develop a system that is somewhat adaptive to changes in DAPI and other types of concentrations, so that, I don't want to use the word errors, it's probably too strong, so that artifacts and things like that of the overall staining process doesn't affect our results. And one of the things that we're going to do as a group, and I'm just going to mention this to put myself on the spot, is release a QA exercise in this so that we can actually look at and compare these techniques, provide some objective data to see how this system performs over time. The second component then to this tissue tethered cytometry is using spatial logic to then break down what fluorophores are present for each individual nucleus. So for the segmented field result that I showed you on the previous slide, we can then treat that nucleus as a hotspot. And then around that hotspot, we can then look at which other analytes are present. We can then break that hotspot up into a pie-shaped wedge or a field, and then we can set up simple rules that say, I want an object to be counted, say, as CD3 positive, if and only if 50% of its cytoplasmic area, as I dilate out from that nucleus, has the CD3 positive analyte. And in this way, we can classify and count multiple analytes per cell, and then we can create a digital spatial map of where those analytes are present, and we have the Cartesian coordinates, so you have a wealth of data then about that information. We call this tissue-tethered cytometry. And it's the basis of what a lot of what Dr. Dimitri shows you. So here's an example. This is taken from one of Sandy's IWIF patients. But this was our, what we called a triple stain with CD45, CD34, and MHC class 2. MHC class 2 being red, 45 being the teal color, and the CD34 being green. And you can see the color classification of the cells as they were detected by NearSight based on that rule-based system. Now the nice thing about this is it gives the pathologist a visual clue as to how well the algorithm is performing. So, and we often talk about this amongst our group. It's a collaboration between machine vision scientists, engineers such as myself, our laboratory staff with the staining protocols and the imaging, and as well as the pathologist to make sure that the data that you're interpreting is actually real and not artifactual. So one of the main drivers, I would say, behind the software that we built was we wanted to make sure we had visualization of all this information. It may be simple visualization, it may just be overlaying regions or color codings or things like that, but we had to make it simple to make sure that the data was accurate. Now in this slide you can also see these yellow circles. The yellow circles are represented by an additional spatial logic layer that we put over top of the slide. And that was to detect portal tracks by finding combinations of cells, MHC class 2 in this case, that clustered together in groups of 5 or more, given they would indicate typically where a portal region may be. So we were then able to then denote with a circle or of a given radius what a portal tract would look like or a potential portal tract area. And so then the pathologists, such as Dr. Dimitris, can then go through and look at it with his eyes and come up with a visual assessment of how well the algorithm is performing. So that assessment and that feedback loop is very, very important for the success of these types of projects. Some of the other features that we had put in to make the workflow go smoothly is regions. So we wanted to make the software system as flexible as possible in terms of seeing an overview, being able to find tissue areas, being able to define regions. In the case of this red object that you see on the screen, it's an exclusion region. So we not only wanted to be able to include areas, but we wanted to be able to exclude areas. If it, perhaps tissue artifact, muscle that was attached to the biopsy, folds, or what have you. So the concept of having multiple region types and to be able to control the workflow was very, very critical. We also have a number of ways, and this is a study that was done with some colleagues in Pittsburgh, Dr. Wiley and his neuropathology team. We also wanted to have a way to combine ways to organize data on a digital whole slide. So in this particular case, Dr. Wiley had these mouse brain tissue microarrays. Each spot, quote unquote, in the TMA was another animal. And we wanted to break those up automatically via grid. We wanted to segment the entire tissue area. We wanted to separate the left hemisphere from the right hemisphere. And then we wanted to quantify the amount of certain DAB labeled stain within those hemispheres. Having the ability to sort of combine modalities and build your own spatial construct layers on top of the system gives us the ability to sort of generate data very, very effectively. And then ultimately that data can then be output as an Excel file or into a CSV that can be pulled into R or some other type of data processing language, and then you can explore that information further. The concept that we called augmented histopathology, this is from Dr. Feng's study, the IWIF. We use this technology, and Jake has shown this in a three-dimensional graph. But we use this technology to look at the ability for the software at a screening time point to predict whether weaning was possible for a given participant or not. We knew from the histopathology reads that portal inflammation, as it stood, was able to separate the two populations. In other words, who could be weaned successfully and who could not be. So we started to explore other morphometric measures. Could we refine that? We also knew that there's a lot of research, obviously, that went into this, and again, Dr. Dimitris has presented on that. But we came up with a number of different metrics. One of them was this concept of a lobular virtual immunosynapse, which is a combination of an MHC presenting cell with a CD45 presenting cell, somewhere out in the lobule. So we use that combination technology of looking at virtual portal tracts and virtual lobular areas that I mentioned before. We also found a marker for an infiltrating macrophage, MAC387. And it's interesting that if you graph those two together, one being on the y-axis, one being on the x-axis, you can sort of see that a virtual fence forms around the green dots, which represent patients that could remain tolerant during the study, versus other people that were outside of that fence or outside of that fenced area. So the concept here was, well, could you provide an artificial intelligence or an augmented machine learning approach to supplement this data? And in fact, if you look at using AI or a traditional feed-forward neural network, probably one of the basic, most well-known types of machine learning concepts possible. They've been around since the 50s. And feeding in this MAC387, CD3, these lobular and portal pairings, as well as the pathology scores, you're able to actually calculate out and classify with an accuracy of about 98%. Therefore, at a screening biopsy time point, again, this would be, I guess, post-spective, so we didn't know the outcomes at the time that this data was calculated. With 98% efficiency on the training set, you could correctly project who was going to be tolerant and who was going to reject. The sensitivity, who we could project as rejectors, was 95%, and the false negatives hovered around 2%. Now, this was all presented in the manuscript in gastroenterology, but the power of this system was it shows you how digital imaging combined with human-graded scores and human interpretation can really potentially add a nice layer of tools or tool sets to the practice of pathology, especially in transplantation and weaning studies going forward. To validate this, we did a number of cross-validation runs. So in our case, since we had a limited training set of about 60 some odd people, we ended up selecting 80% for the training set and randomly then picking 10% for validation and 10% for testing. And then I generated some histograms along the bottom to show the mean accuracy, if you look at the distribution of where those scores kind of fall, was about 78% on the cross-validation, the mean sensitivity of 68%, and the false negatives creep up a little bit to 13%. But still, it shows you the potential of combining these scores together for a much richer tool set. We've since sort of moved this towards automated kidney diagnosis. And again, you showed some good examples of glomerular segmentation and some of the work that you've done with the Banff criteria. But again, extending this concept of multiplexing, you can now start to stain, again, multiple analytes on the same patient, or on the same tissue specimen. And using those same rules of clustering cells that exhibit various types of proteins together, you can say, well, how do I find glomeruli? Well, we can find them by training or looking at a textural representation of the CD34 staining within the glomerulus. You can then use that area as a seed, and then combining that with a collagen 3 stain, I can find the periphery of where that glomerulus is. So now you have recognized a structure, you know where it is with a very high degree of certainty. Once you have that area isolated, you can then look at, are there CD45 positive cells within that structure? Since it's all multiplexed imagery, you can look at where the stains overlap, and then you can count, in this case represented by the yellow circle, where there is a CD45 positive cell within a glomerulus, aka some type of glomerular disease, some type of a G-score, and then extend it along those ranges. The same thing goes for tubulitis, PTC, other types of complications, where we know where the tubules are based on staining. The lab has done a very good job at working out stains for the distal tubules to try and combine them all together. Regardless, you can now analyze and find those tubular areas, co-localize them with the CD45 stain, and then you end up with a false color map, or in this case an analyzed map of where CD45 positive staining exists, where CD34 and CD45 colocalization exists, for examples of PTC or situations where there would be tubulitis. So this is some ongoing work that our group is doing. We're looking at sort of taking this multiplex, making a web-based tool where you can actually take this cellular classification or tissue-tethered cytometry classification onto the web, allowing people to upload images and then run these types of algorithms that were developed. Next is just overall database and workflow software. I think one of the extreme advantages that we've had is we've had an interface that allows us to combine specimen data, templated scoring or template semi-quantitative scoring, as well as pictorial examples and the whole slide digital images together, so that when a system actually produces both a semi-quantitative and a quantitative score, it's all looped together, it's all linked together in one location. I think this has allowed us, especially for looking at longitudinal review, it's been an extremely powerful tool to allow people like Dr. Demetrius to look backwards at what has been done to come up with ideas for segmentation or new types of analytical approaches. The software was developed in Java, and we did this early on with the intent of ultimately making this web accessible as well. Given the prevalence of JavaScript and new HTML technologies out there, it's easy somewhat to move this technology forward. And there's an example of, within the same software that you can use to see the score the quantitative template, you can integrate the whole slide digital image as served up by a web server. We've also now started to move towards automation of the actual scoring template, so that both digitally from extracting data from a biopsy and collapsing it so that you can calculate perhaps automatically a G-score or an I-score, but then also just from a semi-quantitative perspective. So by using rules that can be developed and databased and actually expressed in simple languages, we can develop the individual scoring components, such as I represents this as scored by this template, or the G-score represents something else as scored by the template. And then ultimately, that logic can be moved over to the web. As you can see, I put in a little bit of code because I can't do a presentation without it. But ultimately, you're basically just simplifying the rule to say if the Banff score, in this case, the I-score is greater than or equal to 2, or the T-score is 1, then you have potentially a borderline acute TCMR case. So this automated logic and prototyping it through this type of template will allow us to ultimately move this to the web. Dr. Lupi is doing something very, very similar, and it's nice to see that people are trying to put more objectivity behind how these scores are being developed and making tools that people can actually access and use. The same goes to our website. We call it Site, C-Y-T-E, but basically we needed to develop a viewer that was both format agnostic, aka supported all the major WSI formats, but also supported bright field and fluorescence. Again, so much of our lab's work has been in the fluorescence space, and we needed to make that accessible to the outside world as well. So the system was built so that it could support both image formats. And it's also built so that you can do annotations and that you can provide contextual regions to feed into the analytical systems that exist behind the scenes. And then last, if you actually want to see the system running, it houses the Case of the Month images that are on the TPIS website. Some of you may be familiar with it, but it's one of the early or the original transplant pathology internet sites. It provides information about classification, other types of descriptive material, but the Case of the Month is actually run with the website viewer. It's a work in progress. It's not perfect yet. We're working on mobile interfaces to make it a little easier to use on your phones or on tablets. But the nice thing about it is it's meant to be scalable, and it's meant to sort of include our ability to publish rules, publish digital slides, work with collaborators, and get other data out to the web to make it easily accessible. And then last, I think this is the tip of the iceberg. I think you saw from Dr. Farris's presentation, there's so much potential out there with the generation of this data, both in terms of techniques from a digital perspective on how to do it, in terms of tool sets that you as pathologists, clinicians, researchers can utilize to better improve your ability to generate scores or generate things that are reproducible, much more consistency. But it is the tip of the iceberg, and there's so much more that's underneath the water that we haven't even uncovered yet. So with that, I'd like to thank Dr. Demetrius and the working group for allowing this time to talk about this, as well as the lab and my other colleagues here from the University of Pittsburgh.
So that was great. Does anyone have any questions? That was exciting.
I do for you.
Oh, okay.
And well, I can do it for the podium. I think it's general, and given that I wear many hats, one of them programmer, one of them is junior biostatistician in training. Dr. Hubscher gave a summary of the gigolo data, or the jig data, I should say. I shouldn't say gigolo. One of the things I noticed even in your talk, Brett, is that you talk about kappa scores. So one of the things that we did, and this is more just an impression from the audience in general, we implemented weighted kappa scores. And Stefan presented both, the unweighted and the weighted agreements, where we gave a lesser penalty to none to minimal versus as you move up the scale, the discrepancies would be weighted as a normal factor of 1. Do you, as a group, is that well accepted? I noticed that you didn't do any type of weighting, and you say, well, pathologists, sort of the kappa scores were low. How much of that is just due to discord at the lower levels where there's a none or a minimal and the score is so close together that you don't know?
There probably is. You're exactly right. I mean, there's a lot of ways to look at the data. We also, I kind of glossed over when I presented it, but in that Banff paper, we did report ICCs, the intraclass correlation coefficient, for that too, but they were still pretty, pretty low. They were I think they were a little bit higher than the kappa, but yeah, weighted kappa could be useful. I think they're great statisticians here. I know Dr. O'Leary is very good in statistics, and also Julian Hogan, people who have really great approaches. And so we're starting to look at that too, and we've looked at observer variability and inter-observer variability, and then comparing against the computer morphometry. And yeah, I mean, it's really, I'm probably not the best person to comment on that, but there definitely are a lot of ways.
It was more an observation, because I know some people, I've had feedback where weighted kappa sounds like you were sort of tainting something in someone's favor, but in the case of, you know as pathologists that it's hard to stratify expressions. As humans, we're not built to count pixels. So when there's tiny, tiny bits of stain versus the difference between a none and a minimal is almost a toss-up at that point. So it was just out of curiosity. It was more of an observation than anything.
And I think with the, obviously for continuous data, you can look at, correlation coefficients, which we usually do, the R values, but when you stratify the data into Banff scoring, a lot of times, even though there's a lot of disagreement, the pathologists tend to agree that it's in the same category. So and obviously as you make the data have fewer data categories, you are going to have more agreement. But usually for clinically significant, decisions, severe inflammation versus minimal inflammation, pathologists do much better. So I think my presentation may have come across more discouraging than it actually is. I think that pathologists do tend to do pretty well. Mark Stegall had some interesting data similar yesterday, that he had looked at the Mayo group and that they had some disagreement. Yes, Dr. Demetrius.
Yeah, I think it would just be helpful, like you mentioned early on, to catalog what's going on within the Banff organization altogether and share resources. And that's one of the reasons, I asked Drew to present, to kind of open up the doors of our lab and say, here's what we do, here's what we can share, here's what's going on. And the same thing from other groups, so that the synergy can work to move the field forward faster than it otherwise would. Everybody doesn't have to reinvent the wheel; they can borrow the wheel.
Yeah,
that's a great point. And I mean, there are already really exciting algorithms out. I mean, your platform is amazing, and Julian and our group, we actually the computer scientists we work with have done tons of work in cancer. But now they're very excited. We presented the Banff classification to them, and they were very excited about the Banff classification, how it's more quantitative already than a lot of the cancer literature is. And so they were just, they had done a lot of work on cancer. They had $1 million grants, but actually they were more excited about moving to a different area that already was so quantitative and to use their techniques. And they have a platform for hosting images, and their platform also has morphometry in it, also some algorithms. But I think it'd be great to have a platform people could upload algorithms to have their own new algorithms to it, and eventually we could figure out which ones are better for some things than others, and maybe even have a Banff set of algorithms.
The R community for statistical processing has done that, to an unbelievable degree, in that the repository, you can find anything you need out there. It's been implemented. Now, whether it's stable or it's been tested or that you trust it is another question. But that open source community has done extremely well at providing a collaborative repository for different types of techniques. And yeah, you could do the same thing here, I would imagine.
Any other comments?
Most of the stuff we do, Drew mentioned it during his presentation, is not to say, well, we developed that this is ours, that we're going to kind of gain some kind of advantage out of it. It came more out of frustration for advancement. When you have to deal with a vendor-delivered software, then you've got to deal with all the bureaucracy that goes along with it. It may do close to what you want it to do, but it's not doing exactly what you want it to do. Then you have to negotiate with the vendor and get an upgrade. That might take a year.
$10,000, $20,000.
Yeah, and that's why we wanted to get away from that.
Yeah.
But like Drew said, the integration and the ability to collaborate is there.
Yeah, I agree. I completely agree. It's so much better because we've gone to vendors, and they, for one little alteration, it's usually minimum $10,000, to do something, right?
Well, I think the concept is to produce tools that everyone can use. And we had a nice discussion with Desley, I don't think she's here, but about just even the scoring of steatosis, macro and micro. The web gives you the ability to distribute that tool to everyone. So it's something that we can build. We're not encumbered necessarily by a vendor to put technology out there where there's licensing fees. But it's something that, once it's developed and we continue down that path, it's a tool that everyone can use. So you'll have a website hanging off of allographpath.com, that happens to be the site that we use, where you'll be able to upload an image and, let's say, segment large globule from small globule fat, and we'll see how that works. And it allows you to get the field engaged in providing much more objective and criteria to develop it faster. Because if you do it in a silo, you sort of have your vision of it, and it isn't that our vision is right or wrong, it's just a question of the more ideas, the better off the end product becomes, and the quicker you can probably get there. So there are a lot of low-hanging fruits that our position, I think, as a group, is to get this out in people's hands. Things like basic fibrosis quantification, even based on serious red staining. There's the aspect of polarization that kind of comes into play with that. But just the basic color quantification tools like you have available in ImageJ, to put those up on a web, the Google Drives and the OneDrives and the cloud, for lack of a better term, is so accessible by folks today that setting up a box site where images can be dumped and running a result and being able to log in and see your data probably would help move this field a lot quicker to standardization, I would think. And that may be something that the Banff working group wants to consider, or take up a little more of a little more urgency, because the quicker or the faster you standardize this, I think that ultimately the better your results are going to be in a much quicker time period.
Yeah, we focused on fibrosis quantification. The liver presents a little more difficult problem because it's more like a tree trunk that you don't know when you've got big portal tracts versus small portal tract. Kidney's a little bit easier of a problem. But Drew was also working on simple, even colorimetric quantification, like he showed you on there with inflammatory cells. So if you have it non-overlapping, it's not as good for overlapping signals. But if you have non-overlapping chromogens, then there's going to be a tool to just count the number of CD3 cells per millimeter of biopsy. So we're starting off with kind of low-hanging fruit and putting them into modules so that they're not too complex for the user.
Yeah, I saw Dr. Desley Neal walk in, and we were talking about the steatosis, and we have an approach I presented, but there's definitely more complexity you could put on that, macro versus micro, and I do have a computer scientist who's now working on overlapping steatosis, and it gets to be complicated when you have fat droplets that overlap. Yeah, exactly. So and that's hard.
Ears in the face.
And another group's helping me with liver fibrosis, and like you said, it's so complicated. And I think it also, the thing about the liver fibrosis that's illustrated to me why pathologists still are so important is that sometimes a computer scientist, it's both. It becomes a conversation. You can teach them about, well, not all fibrosis is equal. Viral induced fibrosis is different from steatosis and steatohepatitis induced fibrosis, which is more sinusoidal. So there's a variety of things. So it's a two-way street, and I think pathologists and computer scientists and combined people who do both can help each other, and so I think that's good. Yes, thank you.
Question for the group, and this is related only to liver because I only do liver. In addition, I do cytopathology, so I've been familiar with the digital imaging in cytopathology, and for the last 40 years they've been working on something related to cytopathology, and the most they could come up with is screen slides, and it's only applied to GYN. Come with 3-4 types of cells, L cell, H cell, glandular cells, and still, this has to be verified by pathologists and approved by pathologists. For the liver, what are we talking about? I know it's a very good utility to study pathology and pathologic mechanisms and details, but from clinical point of view, in 20, 30 years, what's the vision? And I want, see what people think about it.
Well, I think what we do today as morphologists is very crude. Okay, so you give, and that's not bad because it's quick. Like I showed in the previous session about the molecular microscope and how they say all the bad things about pathology. But nevertheless, if you go, in my opinion, you go to ground truth, pathology is pretty good and works most of the time and it's damn quick. So the clinician gets a reasonably good answer. But I think what's going to happen in the future, and I've already tested our nephrologist, we're going to give them our estimate of what's going on with the rough band score, and then tomorrow or the next day you're going to get this detailed report with there's 72 CD8 positive cells in the proximal tubules. There's 2.8 monocytes in each glomerulus. So you're going to get a very detailed report like that out from the biopsy, rather than the kind of a crude thing. And with the liver, this is the same thing. I mean, we just give portal information, interface activity, steatosis, but you're going to get quantitative scores on the whole biopsy, which will turn the pathology from semi-quantitative to quantitative, and it'll free the pathologist to do what they do best, to put the whole thing together and conceptualize cause and effect in the biopsy.
The pathology is still your architectural anchor. So myself as the engineer in the group, and what I've learned about it, I've learned from Jake over the last 15, 20 years, but it's still the anchor because everything has spatial relevance. And I think we've learned that, especially working with Alberto and the group, is that even the synaptical pairings came from an idea that looking at some panels that was produced you know, in the gene arrays, it's, oh, isn't that interesting? This signal goes up. Well, maybe we can look for that in the quote-unquote pathology imagery. And I think you're going to see the same type of combination. But as Jake says, I think if one starts to augment the other, and allows the pathologist then to integrate that information on a spatial level, perhaps.
That's very important. That's very important message to emphasize, because now the clinicians, they know about these things, and probably they are expecting miracles to come soon out of these digital pathology.
Well, Banff's already starting out with the DSA. It's a part of the Banff schema for kidney and liver, and you're already integrating serology and morphology, and this is just another aspect, and the gene array will be in there as well. So the more ways you can get to look at a problem, the more certain you are of what you're doing.
Brad, I actually like your concept of augmented intelligence rather than artificial intelligence, because I think that summarizes the discussion of the last 5 minutes of where we might be going with this. I mean, no matter how crude pathology is, there's still a lot of brain work behind it, and we can sit down and look at a lot of things that, a very well-trained genie will pick out as being significant, and I could look at it and say, well, genie, you're smart 99% of the time, but this time, I'm going to override you. So the augmented intelligence thing is going to be good, and I also think, the ability to have a pre-accumulated data before the slide gets to the pathologist, that might be a very good way to look at, all this digital thing and the artificial intelligence concept. So the future is exciting, I think.
Yeah, and I should mention that I kind of got that idea of augmented. I definitely agree with that. I loved it when I saw it. I got that idea from the American Medical Association. So they have a whole website. It's called Augmented Intelligence, and so I think where it came from is that not only pathologists but a variety of clinicians are worried about I guess not worried, maybe not be the right word, but about their job going away because of because dermatologists, they're coming out with ways to recognize nevi or rashes or melanoma and a variety of things. And so that website basically they have a whole website with a lot of articles, and you might try Googling it, and I should have mentioned that in my talk before, but basically they have tons of things there that pointing out different areas where medicine is it's augmented intelligence. You're exactly right, and I think it's a much better term than, because actually I realized that apparently, and it's getting above me, but actually Alexander Lupe doesn't like the term or thinks that it needs to be clarified, this artificial intelligence term too, because it can mean a variety of things to different people. But I think augmented intelligence can also mean a variety of things, but also it has a positive connotation. Yeah. Oh yeah. So I think.
Brad, I have a comment in your talk. Is there a way that we can actually revive your project? And because I really feel that fibrosis is a big issue, especially for kidney pathology. You give nephrologists a 30% score and they panic. And so, especially now that a lot of people are already using different programs, and I know that you use Apereo and built-in algorithms in Apereo. Some people use different programs with their own algorithms. Maybe we can also cross-test what people use and probably survey who's already doing the same thing.
That's a great question, and I think, and Michael Mingle pointed out, maybe we should have a Banff, some Banff algorithms out there eventually, and he mentioned the fibrosis paper that we've done, because it's essentially the first sort of algorithm that's been done as part of a Banff. That was mainly for collagen immunohistochemistry, but it can be tailored to trichrome, and with Julian, we're actually working, the thing about the Apereo, the way I did it in those papers was basically it was case by case. Now Apereo offers an e-slide manager that can do batch analysis, but our computer scientists, and I know you do too, probably have a batch mode where you can upload a number of images and do the fibrosis quantitation on a bunch of cases at once. And it's really much better because, you know, Julian, actually, he and I have been working, he mainly did the work this time. We're going through a set of biopsies now, and we actually submitted an abstract to the next USC CAP meeting, which was due today. Hopefully the reviewers will like that. But I know he's planning on extending that, and also before we do our machine learning. But you know, our computer scientists have a great, what they call histomics TK. And the nice thing about it is it also has color normalization. So, you know, as you know, there can be staining variability from lab to lab of trichrome or a variety of other stains. So their platform actually allows you to sort of normalize the slides before the analysis is done. So I think that this, yeah, you're exactly right. I think we need to come up with another project that can test this, maybe multiple institutions, you know, and to see how we can do this, and hopefully, and connected, maybe we can have nicely curated data with along the lines of what Alexander Lupe is doing, to have a nicely curated data that we can test it on. I think Dr. Andrews has
a question in the back.
Oh, okay. Well, then,
I have a question, more like a comment, and I want to hear your opinion on this. Regardless what algorithm we use or what pathologies you use or how many pathologies you use, a biopsy is just a representation of a little bit of an organ. Right? We frequently see in the liver, especially in my area, is that we do biopsies and then you have 30% steatosis in one, 40 in another one, right? Biopsy in different areas of the liver. How you see this, the use of technology such as MRI or other technology like MRE or MRI in compared with pathology? Because it gives you the concept of the entire organ instead of one single representation.
Well, I guess there definitely can be a lot of sampling error with biopsies. You know, that's one thing we did do with our steatosis project, was work with radiologists to see how well the data was correlating. You know, and definitely, I think, you know, yeah, eventually we could sort of partner with clinicians and have some way to integrate radiology data also. And does that answer your question?
Yeah. Well, I think part of it also is resolution based, right? So, I mean, there was a nice presentation where on Monday when I was here, someone was talking about false coloring, basically half micron resolution data that you can get from, you know, MRIs and micro CTs and things like that. So I think ultimately, you know, the intent would be to incorporate as much of the organ as possible. Jake would probably agree. I don't think the resolution is there yet for the detail that you need. I think, you know, maybe for large level macro changes you could. I mean, similar to the various sonogram type.
Up there?
What's that?
Yeah, you want to?
Yeah,
Yes, yes, okay.
But like as Dr. Farris said, I'm not sure it's there yet. I think, you know, you do see some combination. Jake, you want?
Yeah, so comment here, and then we have two people here. Yes?
if I may ask.
Yes.
Great talk, thank you so much. I'm engineer myself, and I have a few kind of engineering questions too. First of all, can you expand your algorithms, your technique outside of pathology? Let's say for some kind of multivariate analysis, if you can express it in forms of an image, and then apply your algorithms to that?
You could. I mean, again...
Because that's what I had in my mind, like, you know, this can be extended far beyond the pathology analysis.
Yeah, you could. I mean, there's a wealth of information, I think, as you know, on segmentation techniques and data representation techniques and how you sort of visualize things and come to an endpoint. We started looking at some of the T-SNE type approaches, looking at cell populations and things. Frankly, our issue has just been resource limitation. There's just you know, the
You mean
project.
development resource? Or?
Yeah, the project queue it grows longer every single day, and it's hard to, you know, to get through that. But yes, absolutely. I mean, any of these multivariate techniques can be extended to any other domain, or even within this domain to further explore the information. And I think, you know, it's interesting, and that's why I think I use the term living data set. Like, fluorescence gave us the ability to look at multiple analytes on one single location, so that was powerful. But it lets you go back and re-query and keep looking and looking over and over again. So it's conceptually it's the same thing. It's very applicable.
Great. And my second question is very utilitarian. Kind of, how friendly is your API for adopting in some kind of local digital dashboards I might build, or what have you? So essentially, do you expose web API? Or it's some kind of
it's all in C++. From a pure technical perspective, it's all written in C++ right now, just for the utility of pushing around data. Now, I'll be honest with you, that's legacy. That's because this project started many years ago, and even 5, 6, 7, 10 years ago, you needed that level of an API. I think our goal here is to make web-based modules so that you sort of extrapolate out the core. Think of it as an analytical test. So you buy a reagent kit for Key 67 staining, and you open up that kit. It's the same thing here, where we provide a web-based API to submit an image, get a result set, you know, explore it to a certain degree, and then that sort of standardizes.
I would love to be able to consume as a web server, and we can collaborate in that.
Sure.
Absolutely.
Yeah, I mean,
great. Thank you so much.
Right, you want to pass that down?
I actually just wanted to comment about the imaging and putting that together. You know, I think as a clinician, there's several different mechanisms of elastography that are available now. The first iterations of MR elastography required that you outfit your MR equipment with a $50,000 piece of equipment, and then things had to be very different, and so that was less user-friendly as far as dissemination. But there's now a company called Spectral Imaging who actually just has a computer software package that you upload onto your MR machine, and you actually get output of a specific quantitation of fibrosis, a specific quantitation of fat, and a specific quantitation of inflammation. And so I personally think that's a very interesting and important data that's going to need to be integrated with pathology in the future. The big problem with it is it takes 15 minutes of MR time. But what's much more user-friendly is ultrasound elastography, and those pieces of equipment have been downsized so that you can actually wheel them around like a small carry-on bag, and you can take, like we have one that we can take to clinic. I don't know if you do, so a lot of people now have this that you can actually just transport to clinic. You can use it right in your clinic room as almost a fifth LFT. Not that often necessarily, but something that is probably much more helpful when it's negative than when meaning to predict that you have, you know, ongoing stability of the graft than when it changes. But certainly that may be used in the future to prompt biopsies before we have more severe pathology present.
We wrote a review for transplantation. It appeared a few months ago, addressed that specific question. And at least in my opinion now, in the far future, they may merge, but for right now, the sampling error you sacrifice with a biopsy gives you a level of resolution that you're not going to get on imaging techniques. Like, I don't think there's any imaging techniques that can tell you there's a T1 in the kidney or an immunologic synapse in the lobule. So, will it come? Maybe, yeah, will it come. Assaying the whole organ, is it better? Sure, it's better. But the resolution level isn't there yet.
As far as the fibrosis and the FibroScan, actually, we had one of the few FibroScans that were tried in this country and approved by the FDA. It is not, still we don't know the utility for transplant. The transplant liver is a different organ with different vascularity, with different inflammatory components. So you cannot still there needs to be data to apply the FibroScan readings to the transplanted liver. And the other thing about the steatosis and radiology, I mean, we are talking about a small biopsy when we compare radiologic evaluation to a needle core or at most a wedge biopsy. So who's more accurate? And I think still the data is not conclusive. Is radiology more accurate in this instance? Or is it really the biopsy, and you can say the biopsy is a small part, but how accurate is the radiology to predict steatosis and, for that matter, inflammation and fibrosis? I think still, there's a lot of work to be done on these to make sure these apply clinically to patients. It's very early to draw any conclusions about fibrosis and steatosis in CT or MRI studies and apply them to patients. I mean, there's a huge gap still that need to be filled.
Anyone else have any questions or comments?
Question that I'm not sure we know the answer to. So, should we be concerned about platform bias going forward? So now today we have a lot of scanner with analytical capabilities, offering different algorithms, and if you're a lab director, you get emails every day. So is this going to be an issue where we might struggle to translate data from one platform into another?
Go ahead, please.
Yes, I mean, and I think that's, where maybe what Brad said, that the concept of creating a repository or something at an organizational level kind of helps to dilute that. But no, you're absolutely right. I mean, the challenge becomes, how do you count what you count? And I know that, again, I've said it, we didn't design what we've done in Pittsburgh because we wanted to build something or we thought we were smarter. We designed it because we wanted to make sure we had the ability to put the algorithm out there for people. If it has utility, and let other people use it. So it's something that we were able to own cradle to grave and push out as we needed to. But that is a challenge, and I think the only way to eliminate that is, as this field moves forward, is to go into these sort of a repository bank where you have maybe not a Banff approved, maybe it's not the equivalent of Intel Inside or whatever they've marketed, but I think you do have to attack it from that type of perspective, where you have a consortia that sort of stands behind and opens up these algorithms so people can understand them. It's the same parallel that goes on with discussions of black boxes, reproducibility in the AI world. Machine learning is incredibly powerful, but there's a lot of discord now about how reproducible is it. It's based on perhaps your biopsies that you trained with in Minnesota or Toronto versus ours in Pittsburgh versus Emory's, and you're going to get different results. So I think either having a test set or some type of standardized image set, as Dr. Farris mentioned, or even just opening up the methods to some open source or at least to a consortia that can sort of look at them and average out their utility, I think is necessary.
Yeah, I mean, I think it's
not easy because people want to make money is what it comes down to, right? The field,
I mean, money is a big background of everything. I mean, a group like Banff, for example, is what we do is guidelines. So this is probably something, and I don't think we have an answer or something right now. Maybe somebody does. But next year, 2 years, 3 years, we're probably going to get to the point where this becomes like almost everyday thing, and we're going to write. So these are some of the things, even things like thickness of your training set. I mean, do you analyze your serious red or trichrome at 4 microns or 5 microns? And now you're talking about something that is really, really very sensitive, so which is going to pick up, subtle differences like that. So
no, you're absolutely right. And that's why I sort of started the talk with resolution, because we often talk with colleagues about reproducing this, and from a platform perspective, I mean, and Heather knows, conceptually from the technology side of things, counting and measuring spatial relationships, it's not rocket science, as I like to call it, but the data that feeds into it, the resolution and how the images are captured and the focus, it all does affect the ultimate outcome. So I think, yeah, it's a very good point.
The other advantage of making it accessible and make it, we call it maybe Banff crafted. You know, I interact with you on a one-to-one basis, but I say, you know, there's a lot of smarter pathologists than I am. And when you put it out there and they use the tool, they'll tell you, oh, with this is something I would have never thought of. You know, the more input you have, the better the product's going to be.
I think we just came up with a new trademark. So instead of handcrafted, we call it Banff crafted. So I like that actually. It's pretty good. So, I mean, maybe I'm not that well represented in terms of the genomic field, but what ultimately, Alberto, did they do there? I'm not trying to put you on the spot, but there's a number of companies, there's a number of platforms. I mean, they all run. I mean, the algorithms are statistical based, so you do have some fundamental, you know, underpinning of a technique or a method. But I mean, there has to be proprietary, you know, logic in how data is called and how data is ultimately, you know, disseminated from those platforms as well.
There's proprietary platforms, not so much in terms of analytical platforms. So, most of the analytical platforms are actually open source.
So they follow the open source rule.
Absolutely.
Yeah. Okay.
So, but of course, there have been historically huge issues about using different platforms, and in the beginning of the field, huge issues with reproducibility and compatibility of different data sets. I think that things are actually converging as the platforms have evolved, but still batch bias continue to be a huge.
But there's progress. So maybe there's a model that, you know, you can sort of follow.
What's the step? Where are we going? I think just data collection is the next important step.
I do think we're going to start with a survey with the Banff working group. I don't know if Dr. Solis might want to say something.
So another huge issue is the electronic health record and how to integrate this stuff with that. And then the sort of context of use. And you know, in the digital pathology and AI conferences specific to that area, there's been too much emphasis on going totally digital. And so there were only 25, you know, path departments worldwide that had gone totally digital in 2018. Now there are 30. I don't think that's our aim. I think we just want to make it practical.
We were discussing amongst ourselves after you made that comment. I would seriously question the 30.
Yeah.
I don't know of any department that does scanning before primary sign out of all their material. I'm not. Is anybody aware of it?
Do you know any center? Can you name any center that
You
went
are?
completely digital?
Well, Memorial is set up for it.
No, UCACO got up and said, no, we do that afterward. We use the glass for primary sign.
I see. Yeah.
So you,
Yeah, Europe, there are a couple.
Netherlands has two.
I'd like to see them that they scan everything before they sign it
The
out.
Coventry, which is one of those big sites, XG sites, sort of is.
Primarily digital.
A lot of them scan everything, but they use the glass and then keep the archives. All right, well, I stand corrected, but I would doubt whether it's a dozen.
You're still producing glass in the whole.
Yeah, you scan
You
the
have
glass.
to make
So
those to begin with.
So what's
It's a tremendous effort, believe me. We have, what, 5 or 6 scanners, and we have a very small volume, and we're choked. It's a research lab.
Yeah.
It's very hard to do.
Yeah. So I think our effort isn't to get everybody to do it totally for their clinical work end to end, but to just have digital at least on, you know, the educational side or for subsets. And to figure out when you have the data, how you can make it part of the electronic health record.
I think it's a little bit further than that, because Banff is heavily weighted toward clinical trials and studies and improvements. And if we were to do a study in liver now, and we've done this for one study, we can't go back. it's like using the word processor and say, okay, well, that worked okay, but here, go back to this typewriter. The researchers would say, where's that fancy staining you did?
Yeah. Yeah, so we would welcome further input, so we'll be doing the survey. Please take part in that and let us know other ideas, and we'll just keep moving ahead here. But I think this has been a wonderful session. It's probably one of the two best sessions we've had in the meeting. So there. You know. Yeah.
Thank you.
Thank you very much.